Hi everyone, and welcome to another episode of Fried Brain Science. I'm your host, Dr. Brian Fry. Last week we talked about the top 10 poisonous animals. Today we're talking about 10 uses of bioweapons as tools of war or terror. From medieval poisoning of kings to using pepper gas to shut down protests, biological agents have been used by humans for much longer than we may think. The growing fear today is that control of bioweapons will slip out of the hands of state actors and become unpredictable. With the speculations about the origin of COVID-19 virus that has led to the current pandemic, the interest in bioweapons is at an all-time high. In this episode, we're not going to speculate about COVID. Instead, we're going to have a look at existing biological agents that have already been used in warfare or acts of terrorism, or that could easily be used. Bioweapon 1 is the contamination of food or water. This use of biological agents is the oldest form of biowarfare and bioterrorism. We'll focus on Salmonella. This is a type of bacteria often associated with the ingestion of food contaminated by animal feces, human feces, or by eating undercooked chicken. It causes massive diarrhea and can lead to death from extreme fluid loss. As it is commonly found and easily grown, it is a prime one for use as a bioweapon. A particularly high-profile bioterrorism act involving foodborne pathogens was the attempt by the Rajneeshpuram cult in Oregon, USA, to influence a county election by lowering turnout. They contaminated restaurant salad bars with Salmonella bacteria, resulting in a large community outbreak. Luckily, enough of the cult's candidates still did not get sufficient votes to take over the county board and place it under the cult's control. Bioweapon number 2, henipaviruses. These are bat-borne viruses with a high fatality rate, such as Hendra or Nipah. Symptoms may be respiratory, including bleeding into the lungs, or central nervous system effects such as inflammation of the spinal cord or the brain, leading to coma or death. All naturally occurring human cases of Hendra have been infected only after contact with sick horses in Australia. As a result, veterinarians are particularly at risk of contracting the disease. Horses become infected with Hendra after exposure to bodily fluids from an infected flying fox bat. This often happens in the form of urine, feces, or masticated fruit covered in the flying fox's saliva when horses are allowed to graze below roosting sites. Similarly, the biggest outbreaks of Nipah in Asia have been at pig farms, with people infected by the pigs and then human-to-human spread from there on. The risk of use as a bioterrorism agent is due to the potency, potential for lethality, lack of effective treatment options, and high risk of infectivity to other people or hospital staff. Bioweapon 3 is cholera. Natural infections by this bacteria are most often the result of poor sewage or sanitation. The hallmark of the infection is diarrhea on a scale that is almost impossible to comprehend, with up to 10 liters a day coming out, and death the result of this catastrophic dehydration. The disease is particularly dangerous for pregnant women and their fetuses during late pregnancy, as it may cause premature labor and fetal death. As cholera is a highly infectious disease, it has attracted considerable interest as a bioweapon. Historical uses include the World War II Nazis deploying cholera against Russian troops, the Japanese Imperial Army Unit 731 against the Chinese Army, with one event in 1941 causing 10,000 deaths, including 1,700 of Japan's own troops. Unit 731 also used cholera in horrible lethal experiments on prisoners of war. It was also used during South Africa's Civil War. Colonel Walter Basson directed Project Coast from 1981 to 1994, which included the release of cholera into the water supplies of rebel-held areas. Such historical examples show the ease of dissemination and thus how easily cholera can be used in bioterrorism, such as being spread on a salad bar. If you're noticing a theme with diarrhea across the bioterrorism agents mentioned so far, you're not wrong. While Hollywood movies would have us believe bioweapons will cause spectacular consequences like melting skin or instant transformations into another life form, the more likely outcome of a real-world bioterrorism attack will see us defecating ourselves to death. But others may be a bit more spectacular. Bioweapon 4 is ricin. Ricin is a toxin produced by the castor oil plant. The toxin acts by basically starving the body's cells, by preventing the cells from making the proteins they need to function and to grow. The symptoms include vomiting up blood and basically a system-wide shutdown of all the organs in the body. As these plants are readily available and the toxin purification is pretty easy, this makes ricin of particular concern for use in bioterrorism. For example, it has been revealed that Al-Qaeda had produced ricin for use in bioterrorism. Ricin was also used to assassinate Bulgarian defector Georgy Markov in London by the Bulgarian Secret Service, which injected micro-engineered ricin-containing pellets into his leg using an umbrella gun, sort of like Spectre in James Bond. While there are vaccines and antitoxins produced, they are few and far between, and for use by the military only. Bioweapon 5 is Ebola. Ebola is part of a group of hemorrhagic fever viruses called filoviruses. It's not the only one out there, and it's not necessarily the most infectious or the most potent. As a group, they're quite terrifying. While the natural reservoirs are enigmatic, outbreaks occur regularly in Africa, which provides opportunities to obtain material for bioweapon use. These viruses are highly contagious from person-to-person contact, and the mortality rate for some strains may be up to 90%. Vaccines are being developed, but they're quite limited in being very strain-specific. Viral hemorrhagic fevers caused by filovirus such as Marburg and Ebola have been intensively researched for their effectiveness as bioweapons ever since their discovery. But there have been no cases confirmed as yet for their use as bioweapons. However, considering their profound infectivity and extreme lethality, should such an event occur, it would be absolutely devastating. Unlike Ebola, Bioweapon 6 has actually been used. This is tularemia. It's one of the most infectious bacteria, requiring inoculation with or inhalation of as few as 10 organisms to cause death. In the natural world, it's spread by blood-sucking vectors such as ticks, mosquitoes, deer flies, and horseflies. Direct contact with infected animals or carcasses is another mode of transmission. Human-to-human transmission has not been documented yet. Not only are the bacteria incredibly infectious, but they're also very stable. Tyleremia is considered to be an extremely dangerous bioweapon because of its extreme infectivity, ease of dissemination, and substantial capacity to cause illness and death. When inhaled, it leads to lethal pneumonia. During the Cold War, it was weaponized by both the Soviet and US militaries. Tularemia was used as a weapon in World War II by the Soviets against the Germans at the Battle of Stalingrad. Over 10,000 people were infected. However, shifting winds meant that the Soviet troops were exposed as well and accounted for some of the fatalities. The weapon was effective in that it stopped the Nazi advance, but it was ineffective in that there was a large amount of collateral damage to the Soviets' own troops. Bioweapon 7 is botulinum toxin. This bacterial toxin is one of the most potent toxins known, being an extremely strong neurotoxin causing paralysis. This has made it useful for Botox, but also devastating as a bioweapon. 1 gram of botulinum toxin is enough to kill over 1 million people. Luckily, the bacteria cannot be spread from person to person, so patients are not infectious to medical staff. early intervention with the appropriate type-specific antitoxin is essential, but such antitoxins are in very limited supply and largely restricted for military use, and also carry with them a significant risk of allergic shock. The extreme potency and lethality, combined with the ease of production and transport, in addition to flexibility in modes of delivery, aerosol, beverages, or food, means that botulinum toxin is a very likely bioterrorism agent. A number of states named by the U.S. State Department as state sponsors of terrorism have developed or are developing botulinum toxin as a biological weapon. The Shinrikyo religious sect, infamous for releasing sarin gas in the Tokyo subway station in 1995, also developed a number of other biological weapons. This included botulinum toxin, which they tried on multiple separate occasions between 1990 and 1995 to deliver in the Japanese cities of Tokyo and Yokohama. In addition to the ease of production of the toxin using natural bacterial sources, a significant bioterrorism potential exists for the splicing in of the botulinum toxin gene into easy-to-spread enteric bacteria such as Escherichia coli, which would have absolutely catastrophic outcomes. Bioweapon 8 is plague. This is the disease caused by the bacteria Yersinia pestis and can have an up to 100% lethality rate. An early form in history of the use of plague as a bioweapon involved throwing bodies of plague victims over the wall of the Crimean city Kaffa. This also served as an example of the uncontrollable nature of bioweapons, as this outbreak contributed to the spread of the second great European plague pandemic. During World War II and the subsequent Cold War period, both the US and Soviet military developed techniques for aerosolizing plague. The ultimate uncontrollable nature of bioweapons is also demonstrated by a World War II use of plague as a bioweapon. The Japanese Imperial Army infected 10,000 of their own soldiers by accidents during the Zhejiang-Jiangxi campaign, leading to the death of 1,700 of the Japanese troops. The Japanese Army also used plague on a number of other occasions during World War II. This included the airdrop of ceramic bombs containing plague-infected fleas, resulting in thousands of deaths. The uncontrollable nature of bioweapons led to plague outbreaks there continuing for years, with continued loss of life. Due to the high lethality rate, plague is a potential mass casualty agent. It is estimated that the dispersal of 50 kilograms of plague over a city of 5 million would yield 250,000 infections and about 19,000 deaths. But a second wave of patients would cause 500,000 more infections and an additional 100,000 deaths. Bioweapon 9 is anthrax. This is not just a great thrash metal band from the 80s and 90s, but anthrax is also one of the world's most toxic bacteria. The disease is spread by contact with the bacteria spores. Normally, it is spread by contact with the carcasses of infected animals. The likelihood, however, of anthrax use in bioterrorism is very high, and this is due to the ease of cultivation and the stability of the bacteria spores. As such, anthrax has long been investigated for its use as a bioweapon. When weaponized and aerially dispersed, the small size of the spores results in ready penetration into buildings via the air supplies. The production of such bioweapons also comes with tremendous risk, such as the 1979 Soviet outbreak of anthrax outside of a military base where at least 100 civilians were killed, and this was due to anthrax spores escaping due to faulty air filters. Quite reprehensibly, anthrax was also used on prisoners of war by the Japanese Army Unit 731 during World War II, causing thousands of deaths. The Shinrikyo religious sect tried on multiple occasions between 1990 and 1995 to spread anthrax in the Japanese cities of Tokyo and Yokohama. Another example is the post-9/11 bioterrorism attacks that used anthrax spores spread through the mail system to members of the media and to Democratic senators, resulting in 22 infections and 5 deaths. The use of anthrax as a bioweapon or large-scale bioterrorism would be devastating. The release of only 100 kilograms over large, densely populated cities would be expected to cause millions of deaths and an economic cost of tens of billions of dollars. In addition to the immediate effect as a bioweapon, persistent contamination of the delivery sites is very likely based on results obtained during bioweapons testing. For example, anthrax was tested as a bioweapon in 1942 by the British government during World War II on Grunerd Island in Scotland. This resulted in such heavy contamination of the site with the bacteria spores that it was declared dangerous, and this threat persisted until intensive decontamination efforts were finally undertaken. Such persistence of the spore greatly complicates the mitigation strategies when anthrax is used in bioterrorism. Thus, while anthrax is not contagious, you're not going to have person-to-person transmission, the stability of the spores results in a high infectivity rate at the site of dispersal. Bioweapon 10 is smallpox, the true stuff of nightmares. The smallpox virus is one of the most devastatingly infectious diseases, being highly stable and extremely contagious, spreading readily from person to person. Death may occur as quickly as 3 to 4 days after the onset of symptoms. Vaccines are only effective before symptoms occur. After that, the treatment of smallpox is limited to supportive therapy and antibiotics as required for treating secondary bacterial infections. The antibiotics, however, would have no effect on smallpox because it's a virus and antibiotics kill bacteria. There are no proven antiviral agents effective in treating smallpox. As it has been eradicated from the natural world since 1980, vaccine stocks have declined in number and quality. Thus, the vulnerability to a bioterrorist attack using smallpox is extreme. Smallpox throughout history has been one of the most devastatingly effective bioweapons. Examples against the Native Americans by the British military in 1763 and 1764, and by the Confederate soldiers in 1863 against Union soldiers during the American Civil War. The sourcing of smallpox for use in bioterrorism may come from a number of sources, including military, wild, and genetically engineered. The Soviet Union at the height of the Cold War was producing up to 100 tons of smallpox each year, and these stocks have not been fully accounted for. The sourcing of smallpox from the wild would include localities such as the Kolyma River region of Siberia, which in the 1890s buried about 40% of its population after a smallpox outbreak. The bodies have remained intact under a layer of permafrost that is now melting. So, in addition to having the potential to cause outbreak among the local population there, the exposure of smallpox-infected bodies may allow access to starting material for use in bioterrorism. There is also the likelihood of smallpox being genetically engineered in a lab as the full genomic sequence of smallpox is known. And other viruses have been created from scratch in the lab. As viral recombinant technology continues its inevitable advancement, so too is it almost inevitable that smallpox will be made in a lab for terrorism purposes. This is an absolutely terrifying scenario. Okay, so that's a lot to take in. What does this all mean? What are the implications? Well, bioterrorism agents pose a unique risk for several interlocking reasons. Many are easily transmitted from person to person. Most result in a high mortality rate. Typically, they require special tests to diagnose. They may not even be treatable. And this all combines for the potential to cause extreme public panic and social disruption. Crucially, in contrast to the construction of nuclear bombs, the technology needed for bioterrorism agents is not advanced or difficult to procure, as such equipment is routinely used for the production of vaccines and even certain foods or beverages. The small amounts needed due to the high toxicity, such as botulinum toxin, or the ability to rapidly self-replicate, such as Ebola or Smallpox, results in biological agents being extremely economical and easy to use, thus appealing strongly to terrorists. And a particular appeal to terrorists is the lag time between inoculation and detectable effects or infections. The delay due to incubation combined with ready human-to-human spread would also quickly mask the evidence of a bioterrorism inoculation. Another confounding variable is that initially patients may only display symptoms consistent with ordinary colds or flus, but they're actually highly infectious with these exotic diseases during these nondescript early stages. So this puts their close contacts as well as frontline medical staff at extreme risk. Another significant variable is that declining public healthcare infrastructure is a global issue, and in many countries the lack of affordable health insurance is also a feature that will aid in the spread of bioterrorism events, because patients may be delaying seeking medical attention because they simply can't afford the bills. Then, once they do present, the unfamiliarity of the local medical staff to the symptoms of a particular exotic disease will also result in a critical period of time in which the bioterrorism aspect is not detected. So even once the penny has dropped and that people realize there's something nefarious going on, the diagnosis may still be delayed because it might require specialized assays and also containment facilities. And the diagnosis may be impeded if samples are not collected properly before being sent to the specialist labs. So some of these tremendously potent diseases are actually quite fragile once they're outside of the body, and if you have the sample degrade, then you can't diagnose and detect properly what agent is involved. So, for example, during the post-9/11 anthrax bioterrorism attacks in the US, multiple independent sources were providing information to the media, which contradicted each other very tremendously in their reliability and accuracy. As the hospitals and labs initially involved were not experts in such an exotic illness, there was rampant misdiagnosis and false positives and even some false negatives. Then, once an outbreak starts to bloom, due to mass casualties, local medical systems may become overwhelmed and not able to cope with such a high concentration of patients, leading to ineffective quarantine procedures, further spreading the disease. And that in events where there's a very large dispersal area involving multiple local or even international jurisdictions, this will complicate the coordination of response efforts. In addition, due to security concerns or investigational matters, not all information may be shared by the relevant government agencies, which will only add to the confusion and panic. Then comes treatment, whereby local hospitals may not be equipped to deal with highly infectious, lethal exotic diseases, with, yet again, the frontline staff being at extreme risk. To put this fire out will require mass vaccinations, but this is a far from trivial task. For example, military preparedness against bioweapons largely focuses upon vaccinations that are based upon the likely agents to be used as a weapon of war. A case study of this sort of preparedness is the US military for potential anthrax use by Saddam Hussein in the Iraq wars, with military personnel vaccinated and massive amounts of treatment drugs stockpiled. In this case, it was only because of strategic intelligence identifying the threat, including data about where Iraq had obtained anthrax. The reason the intelligence in this particular case was so reliable was that in the years prior, as extensively documented in the United States Congress's Regal report, Iraq legally purchased the anthrax strain used in their weapons program from the American Type Culture Collection in Rockville, Maryland, in addition to obtaining, legally, other pathogens. Iraq eventually stockpiled a massive amount of bioweapons, including over 3,500 kilograms of anthrax and 8 kilograms of botulinum toxin. In contrast to military bioweapons, the use of which are driven by strategic or tactical goals, the aims of bioterrorism is to instill acute, short-term panic and chronic long-term fear. And therefore, bioterrorism may unpredictably utilize any number of live bacteria or viruses or purified biological toxins. Of particular concern regarding bioterrorism is that CRISPR technology for genome editing may not only be used for refined production of biological agents, but may facilitate the development of new forms, such as those with a resistance to antibiotics, antivirals, antitoxins, or have increased virulence, or even novel pathophysiological actions. Therefore, new bioterrorism agents are able to be produced quicker and more effectively than the countermeasures to them are. As with military dissemination of a biowarfare agent, spreading to create an aerosol cloud is the most effective way of delivering a weaponized biological agent across a large target area. However, as the goal is to create panic and instill fear, other modes of delivery are also efficient for smaller target area, including directed at individuals. So the mechanism of delivery may range from the mail, as occurred with the anthrax bioterrorism events in the US post 9/11, to delivery in a shared air traffic location, or in a subway station, or spraying an aerosol over a city, or even infecting food or water supplies. Each of these will be better suited for a particular type of agent, producing a different epidemiological pattern and therefore requiring different treatment or containment strategies. So the agents selected for use in bioterrorism events may be those for which civilian populations are no longer immunized against, such as smallpox, or for those for which commercial vaccines simply aren't available, such as Ebola. In the case of genetically engineered new forms, existing diagnostic kits may fail, and therefore they might not be able to identify them, and so diagnosis may be delayed until more complex, time-consumptive tests are able to be undertaken in these very high security types of labs. And this also means that the treatment options may be very limited, where the antitoxins might not work against that particular strain, or the vaccine may fail if these are genetically engineered strains of bacteria or viruses. So a new vaccine may be months away as a best case scenario, or years, or not even feasible. So as you can see from this episode, biological agents are far and away the most complex public health hazard out there, and that these kinds of bioterrorism events are absolutely chilling to consider. The biological agents covered in this episode are by no means exhaustive. So if there's others that you'd like covered, please let me know in the comments section and I'll research those for a part 2 on this very important topic. As always, please like and subscribe, and we'll see you next week when we cover 10 hugely destructive invasive animals.